Okay, so recording started. Welcome to everybody to this new installation of the International Legume Society webinars. I'm happy to be here today with Syama Chatterton from AgriCanada. Today the topic will be about root rot in pea and lentil. At the end of the webinar, we'll have, as usual, the discussion. If you have a question, if the people present have questions, and also I will share a form for all the present to give us some feedback. So thank you, Syama, and please.
Yes, good morning or good evening, depending on where you are. I'm happy to be here to share some of the research I've been doing on root rot in pea and lentil over the past few years. So I just want to start with acknowledging the funding agencies that have provided funding for this research, and also all of my collaborators. I couldn't fit them all on the title screen as co-authors. This is a very large consortium of researchers that have been working on this problem, and I just like to acknowledge all of the contributions that they have made over the past 10 years. So as we all know, pea and lentils are essential for sustainable agriculture globally. I think it's why we're all here, why we're all interested in legumes. They're very beneficial to agricultural systems. They use less fertilizer because of their nitrogen fixation. They input nitrogen back into the system, again, because of that ability to fix nitrogen. They generally are very water efficient, so they use less water than cereal and oilseed crops. They add crop diversity back into the system, providing that sort of sustainable agricultural framework, and they help break up disease cycles of canola and cereal crops. So there's a really big push to add more pulse crops into agricultural systems because of that benefit and because of their advantage in climate change solutions. So if we look at pulse production in Canada, in Canada, we produce all the pulses: peas, chickpeas, lentils, dry beans, and faba beans, with peas and lentils by far taking up the most acreage. And Canada is the world's largest exporter of field pea and lentil. We're definitely not the world's largest consumer of these crops. And that's why most of what's produced in terms of field pea and lentil is exported out to other countries. And I've shown here kind of a map of Canada, and we can see where these pulse crops are grown. So peas are really grown all over the prairies from Alberta, Saskatchewan, and Manitoba. And then there's also a small production area up in northern Alberta. Lentils are produced primarily in Saskatchewan, with some production occurring in the eastern edge of Alberta. Chickpeas, again, just kind of in this small west and southeast corner of Saskatchewan and Alberta, where it's much drier. And then beans, we have a few pockets of production in Alberta, in Saskatchewan, Manitoba, and then a lot of Ontario. And then we don't actually produce a lot of faba beans, even though it shows that we have kind of the faba bean occurs there. Right now, it's a fairly small acreage, and really what we're finding is that it's starting to replace peas. And the reason why we're looking at faba beans replacing peas is because of the major problem that's occurred in the past 10 years or so, where these pulse root rots, both peas and lentils, really exploded onto the scene in about 2011 and 2012. And in Canada, we have very large acreage fields. And this is a picture taken of a field that I visited in 2014, where we really started to notice this pea root rot problem. And you can't quite see it in the picture, but there is a slope to this field. So this is downslope. This is at the bottom where a lot of water collects. This is higher up. And this area that's green, the producer hadn't planted to peas before, but in all the area where you see that it's yellow and really looking not great, that had a history of pea production. And so we were in 2013 and 2014 were quite wet years across the prairies. And we were really starting to see a lot of fields that were looking like this. So there was a question as to what was going on. And of course, when we pulled up the roots, you can see this complete decay and loss of all root material. So what we found was an unexpected culprit, and this was the first report of the destructive pathogen Aphanomyces eutyches in Saskatchewan in 2012, and then in Alberta when we did the testing, the first report would have occurred in 2013. And this is the very classical symptoms of Aphanomyces eutyches, which kind of separates it from other root rot pathogens. You get this honey brown discoloration of the roots, and you get the shrinking and kind of loss of vigor of the epicaudal. And then it usually stops very abruptly at the green stem. So it's really just a root issue. So after having found it in 2012 and 2013, we then did extensive surveys for the next 4 years, from 2014 to 2017, very systematic and extensive surveys all across the prairies that you can see here on this map. And what we found is that Aphanomyces was really present anywhere where peas are grown. Those are shown by the red pins, and then also where lentils are grown, and those are shown by the yellow pins. So these are all sort of the cumulative positive fields that we found over those 4 years of surveying. And so we can see that even though it was a first report in 2012, 2013, when we went looking for it, it was really present all across the prairies. Then the other issue is that it wasn't just Aphanomyces eutyches. We found a complex of root rot, and most often when we pulled up roots from the field, we don't see them looking like those nice classical Aphanomyces symptoms that I showed you in the last slide. Really what you can see is just this array of symptoms, usually with most of the roots looking like this brown, black discoloration, complete loss of lateral roots. And so this is partly the reason why Aphanomyces may have been missed earlier, because you're pulling out roots and you're not seeing that classical symptoms. You're seeing kind of this mess of symptoms that's really hard to figure out what's going on. So we had done quite extensive kind of culturing work and identification work, and really sort of came up with this list of what I call my gang of root rotters, with our boss of the gang being Aphanomyces eutyches. It usually comes on first. It causes the most damage, but often alongside Aphanomyces, we find a number of different Fusarium species, and I call these the henchmen because they can go out and do damage on their own. They don't need Aphanomyces to kind of help them along. And our primary pathogens there are Fusarium avanaceum and Fusarium solani. And then along with these kind of 3 major players, we have a lot of different associated pathogens. You get a lot of different Pythium species, other Fusarium species besides avanaceum and solani, Rhizoctonia species. Sometimes we find alternaria, sometimes we find black root rot, the Leviopsis. So there's really a lot of different players within this gang of root rotters. Like I said, Aphanomyces is kind of the one that we focus on. And then what really drives these this root rot pathogen complex is wet soil. So when we have wet years, that really saturates the soil, and then also the pea and lentil cropping history. And I'll get into some of those as the presentation progresses. So I just wanted to talk briefly before I start getting into some of our results here is just the issue of measuring disease severity. This is actually one of the most challenging issues working with root rot, is that we're measuring everything on a very kind of subjective visual scale. And because we're working with a complex, at first when I first started working on this, we were just working using kind of an Aphanomyces only symptom scale. But it doesn't really work when you start adding these other pathogens in the mix. So we had to come up with a scale that tried to combine, what we were seeing from Aphanomyces, Fusarium, Pythiums, everything combined. So we developed this 1 to 7 scale, with 1 being healthy, 2 just starting to see a little bit of root rot, and then all the way up to 7 where the plants are dead. And so generally we kind of think of 1 to 2 as the roots still being healthy, disease severity here of 3 to 4. We'd call that a moderate severity, and then anything above 4, so 5, 6, 7, we can see here that the roots are really rotted. And then we can see that you're starting to get that combination of Aphanomyces here. You can see that epicaudal being shrunken and a loss of vigor, along with that blackening that's caused by Fusarium. So rating disease severity is definitely a challenge. It is subjective, and this issue will kind of play out throughout the rest of the presentation when I start getting into some results and how we can correlate things back to disease severity when we're using a categorical rating scale. So first, I just want to talk about the problem in Canada, what we found over the past 10 years with surveys. And this is really looking at Aphanomyces root rot. So what we do is collect, or my colleague, Mike Harding at with Alberta Agriculture, he goes out and does the surveys and then brings the roots back to us. We do the disease severity rating, and then we keep the roots for testing for Aphanomyces. So this is just showing what the percentage of fields that have tested positive for Aphanomyces over the past 10 years. And that's in the green bars here. And then I've superimposed precipitation levels from April to July with the line graphs. And just to look at that, really Aphanomyces root rot tends to follow the water. So we had wet years in 2014, 2016, 2020, 2022 and 2023 were sort of moderate for wet years. And we can see that prevalence is kind of going up and down and following that water. In 2018 and 2019, we had two dry years back to back, and producers actually stopped paying attention to Aphanomyces. It kind of fell off the map, and then it had a nice surprise in 2020, which was quite a wet year. We say, no, it's still there, about 70% of the fields we could find it in. And then we can see in that another dry year, 2021, it drops off quite drastically down to only about 20% of the fields, and then back up to sort of 50 to 60%. So, we look at that on average across the years, we're looking at about 50% of fields that are surveyed were testing positive for Aphanomyces. A little bit of a different story for lentil. First, we haven't been doing surveys every year. Lentil has a smaller acreage in Alberta, so we don't surveys aren't done every year, but we can really again see that lentils really, for Aphanomyces root rot and lentils, were really following those water patterns. So again, 2016 and 2020 were fairly wet years, and you can see that about 50 to 70% of lentil fields tested positive, whereas our dry years in 2015 and 2017, it was very low amounts of, very low percentage of lentil fields that actually tested positive. And then 2023 was kind of in between, and we can see that we are kind of sitting at that 40 to 50% of fields that are positive for Aphanomyces. So the big question is, has root rot impacted pulse production in Canada? It's a big industry here, but it's been very hard to sort of get an economic sense of what's the economic value of this disease. So I went and looked at just kind of past collected data from the past 20 years from Statistics Canada, which publishes kind of all the crop production stats and just graphed them out. And so we can see that pea production in Alberta was very low in 2004. And this is really like the Cinderella story of pulses, is that they started out low and they really had this surge of growth and interest from 2004 up to about 2016. And we saw kind of it hit max production in Alberta at about 700,000 harvested hectares. And then after that, we do start to see this decline in the past 8 years. And now we're kind of sitting at about 400,000 hectares that are harvested. And then if we look at average yields across that time, we can see that the yields really haven't changed a lot, just slightly above 2,500 kilograms per hectare in 2004, and maybe a slight decline to about 2024. But those yields have really kind of stayed at the same place. If we look at pea production in Saskatchewan, which is, they produce more peas than we do in Alberta, we can see a bit of a different trend, and that's that it's been kind of this gradual decline in pea production acres over the past 20 years. So it's kind of starting above 1 million harvested hectares, and then now at 2024, we're sitting at about 800,000 harvested hectares annually. And then when we look at the average annual yields, we can see these are slightly lower than what we see in Alberta, and those have also stayed very flat over the past 20 years. So it's been really difficult, I guess, to determine if Aphanomyces root rot has started to affect pea production. We are for sure seeing decline in pulse acreage, but this could be also due to a combination of factors. Commodity prices, we know, are the biggest drivers of decisions of producers to plant a crop. And also there's been some drought in these years, and you can see 2021 was quite a drought year, and you can see how much production dropped in that year because of drought. But certainly, anecdotally, when we visited fields and we've seen fields that have been really hard hit by Aphanomyces, we talked to producers, we know that producers have removed peas and lentils from their rotation because of their issues with root rot. And then if you remember from one of my first slides where it said that only 10% of fields in Canada or cropped land is cropped to pulses. So there's a lot of virgin pulse fields that haven't had a pea or lentil crop. So what we're seeing is that maybe as we're removing some fields from pea and lentil production, some fields are taking up pea and lentil production where they haven't had peas before. So it's maybe staying stable in that sense. But we are seeing a decline in pea production. Whether it's because of root rots, it's hard to say, but absolutely root rots are currently #1 constraint to pulse acreage expansion and are top of mind for all pulse producers. So how do we stop this gang of root rotters and give peas and lentils a chance? This is very, very important right now because those disease plants no longer provide that nitrogen benefit, and the removal of pulses from rotation are negating any of those climate change solutions that we really desperately need from pulse crop production right now. So what's our path for root rot management? A few years ago, AFC, Agriculture and Agri-Food Canada, we put together kind of a root rot research network, and the scientists all sat together and sort of came up with what our research path or vision is for root rot management. And this is one of the 3 major points that we came up with. The first one is developing soil testing, or developing a method for reliable inoculum quantification so that producers know what's in their field before they plant, and really focusing on this idea of catching fields early so that if there's any management strategies that can be applied, they can maybe be applied to the small patches that show up early before the disease spreads to their whole field. And then once you have your soil test and you know whether or not you have Aphanomyces, then we can stack that with in-field management. And so a lot of research is needed in this area, looking at how long do we have, interval period do we need between pea and lentil crops? Are there any chemical and cultural control methods that work? Are there any agronomic practices like seeding date, fertilization, things like that, that will affect pea root rot? And then, of course, all of that needs to be built on this foundation of having partially resistant cultivars. And along with that, you know, we need to know how these cultivars are working. Are they reducing inoculum levels? How can we deploy? Can we deploy partially resistant cultivars in fields that are infested, or do we have to be careful of not overloading that partial resistance with really high inoculum levels? And so, of course, then that ties back into knowing what's in your soil. So for today, sort of for the research part of this presentation, I'm going to focus primarily on research I've been doing on soil testing and some on in-field management. I'm not going to talk much about partially resistant cultivars, mostly because I'm a pathologist and not a breeder. And so I think there's other talks coming up later in the ILS series on kind of breeding for Aphanomyces, and because this is an area that we've just started working on in my lab. So I'm going to talk about soil test. And like I said, I think this really has to be the foundation of our management practices. Producers need to know whether they should be planting a pea or a lentil in this field. So this is actually a picture of my husband, who's a serial agronomist, but he plants some field trials for me. This is what a field that we're putting a trial in, but this is sort of the question that every producer faces every spring is, should I be planting peas or lentils in this field, and how do I know whether it's safe? And then the problem is, I think most of us that are familiar with Aphanomyces eutyches, the problem is that it stays dormant in the soil. So we have this nice bed of soil just ready for a crop, but what's lurking inside underneath are these oospores that are sitting there lying dormant. So we have our sleeping oospores that are dreaming of a pea or lentil crop. And so as soon as we put a pea or lentil crop into that field, and then we add water in those wet years, that awakens the sleeping beast, and the oospores produce these zoospores. Thousands of zoospores can be produced from one oospore, and then the zoospores are what go and infect the roots. So we can't tell whether there's these sleeping oospores in the soil. And so that's really what we want to be testing for. And the big question is, can we test for Phantomyces, and more specifically, can we quantify the amount of inoculum that's in the soil? And right now, sort of the recommendation that are given to producers is to really be taking good detailed notes and records of their fields, and for them to think about how their fields did the last time they planted peas or lentils. And that generally would be about 3 to 4 years prior, because they're usually on a 1 in 4 rotation. So we have a couple of different kind of field scenarios here, and we're like, okay, think back. 2020 was a wet year here. What did my field look like in 2020? You know, if it was like, well, if it was lush and green, no hints of yellow at all, then you are safe, should be safe to plant peas or lentils in 2025. If your field looked like this, you can see very obvious missing patches of plants, yellowing, yellowing plants, very poor canopy closure, very obvious signs of root rot, then you say, no, do not plant peas in 2025. And then we encounter a lot of fields where you can just see these yellowing patches start to show up, usually along water tracks. So here's the low spot in the field. This one, you see nice water kind of moves along here, and that's where we see the root rot. These are the fields that are a lot harder, first, to remember how your yields did, because they actually might have yielded very nicely, and to remember where those yellow spots actually occurred. So those ones are harder to make decisions on, and those are the ones that we recommend for soil testing. What happens when a producer puts their plants, peas in a field, they go to look at their pea crop, and we see this early yellowing, we see the canopies not filling out, and you know, then they realize, okay, I should not have put peas and lentils into this field this year. And then me as the researcher then gets to yell at them, and I can yell at them because this is my husband in this tractor, saying, well, I told you to soil test. Now, the problem with soil testing is that Canada, our pea and lentil growing region, like I said, is very vast. We kind of have about 1,200 kilometers from one end to the other of the pea and lentil growing region. And we also have a lot of different soil types. So our soils are categorized into a black soil zone, which have high organic matter and fairly wet. We have a dark brown soil zone, which tends to be kind of a heavier clay soil and kind of moderate in precipitation. And then we have a brown soil zone. It's called the Palliser Triangle here, which tends to be fairly dry, low organic matter. Soils can either be heavy clay or they can be kind of a sandy loam. And then superimposed on these kind of soil zones are also all the soil types. So we can go anywhere from a very heavy clay soil up here. Like I said, those tend to occur in the dark brown soil zones. You have the silty loam, which are maybe more characteristic of the black soil zones. And then we have our sandy loams, which you tend to find more in the brown soil zones. But the problem is that even though I can say that's where the soil textures kind of land, when you start looking at different soil textures in these different soil zones, you can really get the whole range of soil types. So what we wanted to do then was look at how soil texture and soil type affected quantification from soils. So these are my 2 technicians that worked on this project. And what we did is we collected soil from the black, dark brown, brown soil zones, either sterilized them or kept them autoclaved, added oospores at different levels, removed aliquots for DNA extraction in qPCR or ddPCR, and then measured the ITS copy number. This is often used for a Phantomyces quantification because there's multiple copy numbers. The problem is the copy number can actually be very variable per isolate. So it can range from 20 copy numbers per isolate. Some isolates will have more than 200. So on average, you end up with about 180 ITS copies per 1 oospore. And this makes it a little bit difficult for quantification. And then along with those soils, then we also did greenhouse trials for disease severity. And the idea was to try to then relate our ITS copy numbers to the ooze spore concentration, which then we have related to disease severity, is to see how well we could actually quantify ooze spores in the soil, and also to see how soil type and soil texture affects that relationship between ooze spore concentration and disease severity. So this was a greenhouse study, and then on top of that, we wanted to say, well, if we have field soils and we do the exact same thing, what are the results there? So these ooze spore dose experiments are really cool because they give you some really nice pictures. This is one of my favorite pictures that we've taken, and because it really illustrates what we're looking at. And what we can see is that at ooze spores doses below 100, we really don't get a lot of disease. And then as soon as you hit that 100 ooze spores per gram of soil, it's like a switch turns on and you start getting disease. And then as you increase those ooze spores, you can see that kind of past 500, it doesn't really make a difference. Everything is dead. We have maximum disease levels. So that shows really nicely in a picture, but then when we start actually looking like what does the data look like and graphing this out, we can see that the relationship really changes depending on the soil type and soil zone and soil texture. And so this is the clay loam soil here. I think this is from the black soil zone, shows a very nice relationship, very low disease below 100, and then you see it progress above 100. Contrast that with the silty loam soil here, and we can see that it's not as clear cut of a relationship there, and that maybe we saw a lot of disease before we even had ooze spores, indicating that there was other things in that soil. So this is sort of the first kind of unexpected result where we're like, okay, well, we can't expect that there's a linear relationship between ooze spore dose and disease severity in all different soil types. Then when we looked at, well, how well can we quantify the DNA from this soil? So here on the y-axis, I have the amount of ooze spores that we put into the soil. And then here is how many we measured back using either quantitative PCR or digital droplet PCR. And we can see that for the most part, for all these different soil zones and soil types, there is a fairly good linear relationship. So it means, okay, well, we can, even if our relationship between ooze spore dose and disease severity is a little bit wonky, we can at least measure those ooze spores, except for some soils, like example here, we can see that we are underestimating the amount of ooze spores in this soil type, whereas other soils showed a better, like the silty loam here from the brown soil zone showed a better relationship. Okay, so that was just a big study. You can summarize it in 3 sentences, really, and that's that soil types differed in disease severity response to the various ooze spore levels. But for most soil types, Aphanomyces root rot did not develop at ooze spore levels below 100 grams per soil. And that had been published in the literature from a couple different researchers, I think both in France and in the US. So that confirms that, yes, we're kind of working with a threshold dose of 100 ooze spores per gram of soil. And that threshold dose can vary between different soils types, but for the most part, it holds true. And there was fortunately a linear relationship between the Aphanomyces ITS DNA that was measured in soil and the amount of ooze spores that we put in. But like I said, in some soil types, the DNA measurement did underestimate the number of ooze spores. So this was a good sort of base to then look at, okay, well, we have this nice greenhouse kind of lab-based relationships. Now let's see how this translates to the field. So what we did was collect soils from about 100 fields that had unknown root rot history. We do a bioassay with them. So we just plant up those soils in pots, put some pea plants in them, look at the disease severity. And so we did measured the ITS copies in the soil before we planted the bioassay, and then we measured them again in the soil after the bioassay, and then also tried to relate that to disease severity. So what we can see here is when we convert those ITS copies to an estimated number of cells per gram of soil and compare that to the disease severity rating, we can see that there's a very poor relationship in field soils between our DNA quantification and disease severity rating, and that for the most part, all of the soils had inoculum levels that were underneath that threshold of 100 oospores per gram of soil. So even with these soils here that cause disease ratings of 5, 6, and 7, so very high disease, you know, we're still coming up with under 100 oospores per gram of soil in that starting material. But when you look at it after the bioassay, we can see that numbers have really increased. So they've increased about 100-fold from those pre-bioassay levels. So I mean, that's not surprising because the Phantomyces is doing its thing. The oospores are germinating, they're producing zoospores, mycelia. So we're detecting things other than the oospores in the soil. But this sort of relationship in the pre-plant soils was very puzzling. So then what we did next was look at this in-depth study of 8 fields. For this one, we tried to choose 8 fields that had a different disease severity levels. So we had some prior knowledge of these fields. And then set up these little bioassays where we put a whole bunch of seeds in a small amount of soil, and then extract the DNA from the roots and the soil every 2 days for 32 days. So this was a lot of work that Shamila did, having to extract DNA from roots and soil every other day. And then we looked at disease severity at the end, and then quantified the Phantomyces in the roots and the soil over that time period. So not going to show you what the results look like for all 8 fields, but this is just an example of what we found. So our pre-plant levels would have been 52 ITS copies per gram of soil. So really that's well below our threshold of 100 oospores per gram of soil, because 52 ITS copies, remember there's approximately 180 ITS copies per oospore. So I mean, we're saying here that, you know, we're maybe less than 1 oospore per gram of soil to start. But then when we start following that trend every 2 days, we can see a surge. It starts to increase. The amount increases at 5 days, increases more at 7 and 9 days, and then stayed relatively constant over the next couple weeks. And then it started to decline, the ITS copies that we could quantify from this soil, and started to decline by the time we did our disease severity here at about 32 days. So this was, I guess, what we were expecting. We expected to see the surge. What we did not expect to see was this decline at the end. There's always a question of whether, when you're doing DNA quantification, if you're picking up, you know, dead material, dead mycelia, dead zoospores, all these other things that could be in the soil. And so really, when we're seeing this conversion of, you know, oospores to mycelia and zoospores are infecting the roots, and then conversion back to oospores, we can start to see that our ability to quantify is dropping off towards the end. Yeah, so then what we did with this was then move on to a much larger study over the past 2 years, where we put out a call for producers to collect samples from their fields for us to test in these soil-based assays. So these are the instructions that were sent out to producers to choose 2 sites within their fields, either low or high yielding sites, low pooling sites, anywhere where they notice yellowing, and to collect soil from a good site and a poor site within their field, send us the soils, and then we are going to test them. And so we tested them in that same way that I showed before, set up these bioassays, and then rather than doing every 2 days, for 32 days, we just focused on that pre-plant, the 5, 7, and 9 days after planting, because that's really where we saw that surge in being able to quantify a Phantomyces. And then we also did a 28-day bioassay and then compared that with in-field mid-season. And so this is a bit complicated, what we found. I'll try to walk you through it. So again, we have our ITS copies here and our disease severity along the axis here. And this is for soil quantification. What we found is with the pre-plant soils in the blue line here, that very poor quantification, a fairly low R-squared value of 0.22. It increases as we move up to the 5 days, and then the 7 days after planting here in the green line, and the purple line here, which is 9 days after planting, we can see that that relationship, that correlation is starting to look a little bit better, but we're still sitting at about a 0.62 R-squared value. And if we look at the root quantification, we see very similar. So fairly poor quantification or correlation at that 5 days, and then it increases at 7 and increases at 9 days. And if you break that down further to look at what was happening by disease severity category, things start to make a little more sense. So if we look at the soils that had a rating of 1 and 2, so that would be kind of on here on the scale, very low disease severity, we can see that that amount, that quantification ability does not change at all over the pre-plant up to 9 days. The moderately diseased plants that had the rating of 2 to 4, we can see that it starts out the same, the pre-plant starts out the same, but we can see that it does increase over those 9 days. And then the very highly diseased plants, the disease severity rating above 4, you can see that first that pre-plant level is actually much higher, and that you get that increase over time. And then the root quantification follows the same pattern, except for root quantification numbers are much higher. So what we're looking at now is trying to develop a model based on this idea that the levels in the soil change or don't change depending on inoculum levels. So that is a work in progress, and just some sort of questions, I guess, on why we're seeing this very poor pre-plant soil quantification. Are the ooze spores with their very thick walls, are they resistant to DNA extraction techniques? Can we maybe look at techniques to try to concentrate the ooze spores from the soil prior to extraction? We have played around with that a bit, and so far nothing has worked very well. And so what we're proposing is this hybrid technique between a soil bait and direct DNA quantification based on this 5 to 9 days after planting surge that we see in Aphanomyces quantification. But the big question is that amplitude of the surge, is that related to the initial concentration of dormant ooze spores? And so we're trying to do some modeling to figure that out, and then hope to continue kind of testing 50 to 60 fields per year over the next couple of years to build a larger database, because models work better with the more data that you have in them. So I'm going to switch over now, so you don't have too much time left, to talk about management options. This is really a field trial fails, kind of giving a spoiler here. Over the past 10 years. The difficulty with running these management trials is that we often have to run them in infested fields. We can't do kind of an uninoculated or disease-free control because it takes so long for that Aphanomyces to build up in the soil. So I'm just going to set the animation here and show you everything we've tried over the past 10 years that have not had success with. Okay, I don't know if everyone could catch that in that short amount of time, but basically we've tried a lot of different things. And the only thing that we've had any partial successes with, this is usually where we'll see a small yield increase, but often without a corresponding decrease in disease severity, was when we had peas directly after a fava bean or oat crop had higher yield at some locations. Gypsum increased yield in one location. Intercropping with mustard sometimes increased pea yield. Again, it really depended on location. And then something we've just started to look at is sort of cultivar genotype selection. Some of the genotypes with some partial resistance combined with the seed treatment are showing some promise. But that's over the past 10 years of a huge number of field trials. This is really where we've had some successes. And we are looking a little more closely into this use of gypsum and then fava bean or oat as a preceding crop. Okay, the one thing that we know does work is crop rotation. And crop rotation works in 2 different ways, both for prevention and avoidance. So I'm going to try to touch on both of those in the little bit of time I have left. So this is the mantra of every pathologist is rotation, rotation, rotation. And so the question is, like, how often can we be putting susceptible pulse crops into a field before you get root rot? And it does depend a lot on how wet it was when you had those peas or lentils in your field. And I think it depends a lot on luck, because we talked to some producers that have followed the same rotation as their neighbors, and they have it and their neighbors don't. And then this was a study that was done by Kuei Liu at AFC and Swift Current. He was not focusing on root rot. He was focusing on agronomy of pulse-intensive rotations. And then just happened in 2022, he noticed that he was getting a lot of root rot in his pea crops. And so I was like, oh, perfect for a pathologist. Let's see what's going on. So in 2023, after his wheat crop, we collected soil to see what's happening. And basically, this is just a series of rotations with a whole bunch of different intervals of pea and lentils. And I don't want to get into the specifics, kind of show you what I'm talking about on this slide with our results. So we collected soil from his rotations and then did a bioassay with pea, and we did a bioassay with lentil to see how much root rot was there. We can see these top 3 rotations that had no peas or lentils in them at all were actually starting to show a little bit of moderate root rot, and this was all due to fusarium. The next 4 rotations next 3 rotations that didn't have any peas, only had lentils at various intervals. We're starting to show a little bit of root rot in the peas, but really not showing a lot in the lentils. And then these last rotations here that had peas 2 times in the past 8 years, all the way up to 6 times in the past 8 years, we can see that that is where root rot really started to develop, and we're into that moderate or that severe root rot rating category. And then we looked at Aphanomyces presence. There was Aphanomyces in all of these plots here with that history of pea cropping. So really it's showing that 2 crops of peas in 8 years were enough to get Aphanomyces root rot going in this location. I should mention it was bad luck. Where he had peas, the 2 years of peas, this rotation here was in 2016 and 2020, which are both very wet years. So it just shows how kind of that wetness ties in with that rotation interval. Okay, so then the other part of root rot of crop rotation is avoidance. So you have root rot. Now what? Now you have to rotate away from peas and lentils. And then a big question has been, well, how long does it take for those oospores to break down in the soil? From some tests that were done in 2016 after a pea and lentil crop by a colleague at Swift Current, what they found was that the amount of inoculum left behind in soils after a wet year in 2016 was about 1,000 oospores per gram of soil in a pea field and about 100 oospores per gram of soil in lentil fields. So that's sort of our baseline of inoculum that we are working with after a pea crop or a lentil crop. So what we did was we tried to, we inoculated roots with Aphanomyces, got some really good diseased roots, put them in these cages, planted them into the ground. So it looks like this. And then with the idea is that we'd follow disease progression over 3 years or until, which is about as many cages as we could get planted in this field. So this was done by 3 technicians here. And so basically every 3 to 6 months, we pull out these cages and look at, okay, what are the oospore levels in the soil remaining in these cages, and what happened with the disease severity. So we can see when we put these in the soil, we were targeting about 1,000 oospores per gram of soil, and that's what we had, so that was good. We put them in November 2021, and then we saw a fairly big decline in that first year from 1,000 down to 100 oospores per gram of soil. And again, that's kind of what our threshold is. And it stayed at that since May 2022. Then if we follow the disease severity, I can't really make sense of these results. It's just sort of going up all over the place. It started pretty low, even though we had 1,000 oospores per gram of soil. So this is right at burial. And then it actually increased the next 2 time points. And then we've seen it kind of fluctuate, but it stayed fairly high. So over the 2 years of the experiment. And we have 2024 data. We just haven't processed it yet. So this was, maybe showing that oospores rapid decline, and then they reach the sort of stable period where you don't really get a change in their inoculum levels. So yeah, that's a big question. What is happening with oospores in soil? Are we just not able to is it the techniques that just can't accurately quantify them? Do oospores numbers actually rapidly decline in soil? Maybe we can produce these lab and greenhouse threshold values saying it's 100 oospores per gram of soil, but is it actually kind of our field-based threshold and active spore load in the soil that we can measure might actually be lower? Okay, so how long then do you have to wait to grow peas and lentils again? And this is the last experiment I'm going to talk about, gone a little bit over time. And so we did this fairly large rotation study across 6 different locations, looking at how long of rotation we needed with the cereal and canola, and if we included an alternate pulse crop in those locations, how did that affect pea root rot? So we had chickpea and Tabor and Swift Current as our alternate pulse crop, faba bean in Lacombe and Saskatoon, and then soybean in Redvers and Morden. And I'm only going to show you data from Tabor because every location basically looked the same. Again, this is a lot of data to show, not very much. And really what we're looking at is that our disease severity index with a 1-year, 2-year, 3-year, 4-year, and 6-year break really we did not see a reduction in that disease severity index over those 6 years. The 7th year that we've had this rotation in, we've started to see, as the first year we've seen a decline in that disease severity index. And then if we follow pea yields, you just see they're kind of all over the place. We had really good yields in 2019, but that's when we had kind of a moderate moisture. It wasn't too dry and it wasn't too wet, and we actually got really good pea yields. And the years where we had very, very wet conditions, we had horrendous pea yields, 150. This is a 3-year break, 70. 6-year break, we're still only at about 50. That was actually because it was too dry. And then in 2024, we're starting to see a little bit of recovery in yields with that reduction in disease severity. But keep in mind that our provincial yield average is about 2,500 kilograms per hectare. And so we're sitting well below that in this infested field. So if we summarize what happened out of all those 6 sites, 1 site actually did recover after about 5 years, and that was in the black soil zone. All the rest are going to take longer than 7 years because we've only hit the 7 years now. But the yield loss relationship to disease severity really was not clear. I know I showed it very, very quickly, but the yields are just we're all over the place. It seemed to be more dependent on, that those wet conditions with Aphanomyces is the most detrimental to yield. But even under those drought conditions, we still had high root rot severity, and we still saw some yield loss. I didn't get into it, but the soybean, faba bean, or chickpea really didn't impact root rot severity in either direction. So it means it wasn't contributing to more root rot, to more inoculum, but it also didn't help. They also didn't help reduce the inoculum. The only thing we saw, like I mentioned in an earlier slide, was that peas after faba bean in some locations actually did produce a better yield, and we are looking further into why that occurred. So I think this rotation study just really reinforces the idea how important a soil test is, because it needs to work over prolonged intervals. A producer that's decided to take peas or lentils out of production, they need to know when they can go back into production. And so how the heck are they supposed to know if they should plant peas or lentils there? Right now, I feel like it's a little more of a guess than an absolute science. And we are trying to get that soil test down for them to be able to make those decisions. So in the end, I think our path for root rot management is not a nice, straightforward path. It's long, it's arduous, it's circuitous. I love hiking in the Rockies, and we sometimes go on what we call these scree slogs. This is what it feels like sometimes in this Aphanomyces root rot research. You take 1 step forward on the scree and you slide 2 steps back. And so I think that's where we're on. Right now, we're not at the bottom. We've made some progress. We're making our way up to the top of this mountain. And so just in summary again, I think we're making progress on the soil testing. We're making progress on infield management. And I did not talk a lot about partially resistant cultivars today, but there's lots of other researchers that are actively studying that. And then just to end on a more hopeful note, say all aboard the peas train in 2024. There's a lot of interest in root rot research in Canada. We're seeing it really take off. There's 3 big projects that have just started this year. One is part of the Pulse cluster, where we're looking at a lot more into the lentils. I didn't talk much about lentils today, so we're looking a lot more at lentils and trying to work on that improved diagnostics and coupled with agronomics. Big project, Genome Canada, that was just funded, and that's really focusing on breeding and genomics and genomic selection for partial resistance. And then the Saskatchewan Research Initiative, that is a very large consortium of researchers. They're looking at all sorts of different things. So I think we're making progress. There's lots of exciting research happening. And I'll just end this. This is what fall in the Rockies look like. This was just taken last weekend. We have these large trees that turn golden yellow, and everyone gets out to visit them. And I thought I'd just end off the presentation with that. Sorry, I went a little overtime.
No, you were perfect, and very interesting presentation. So thank you again. People in the chat is virtually uploading. So thank you again. It was a very interesting presentation. Just as an organizational note, in the chat, I put a link for a feedback form. So please take a moment to fill it. And now we have time for questions. If you have any, you can directly unmute your mic if you want to talk, or write in the chat, and I will read the questions. Or okay, if the audience is okay, let me see. Okay, so thank you for the presentation. I'm interested about natural variation. If you work with different cultivars, do you ever notice high amplitude variation for severity of the disease depending on the genotype?
Oh, that's a good question. Yes, we have just started working with different cultivars and different genotypes in the field. So far, I would say there's not a huge variation in severity depending on the genotype. But yeah, it's a difficult question, because we've also just started this very large screening project where we put about 400 lines in the field, and then we do see kind of a nice normal curve for variation. So there is some variation of that disease severity, but when we look at cultivars specifically, all our cultivars tend to have the same reaction. They're all equally susceptible. When we're looking at kind of germplasm accession lines collected from like globally, we are seeing that there is some variation in disease severity, but most of them tend to be on the high end.
Yeah, that will require then a long path of introgressing the resistances into commercially available cultivars. Let's raise the hand and then a couple of questions from the chat. So Lars.
Thanks, Syama, for a very nice presentation. Really enjoyed that. I had interest in the rotational studies that you did. Do you believe that the lack of reduction in oospores per gram of soil is as a consequence of other post crops in the rotation tolerating the disease, or is it because they can persist for long periods of time in the soil?
Yeah, I think it's because the oospores can persist for a long time in the soil. I know I was trying to show a lot today, and what we had done in that study is that we had actually collected the roots of the faba bean, the chickpea, and the soybean and tested them for Aphanomyces. And faba bean and soybean didn't support Aphanomyces at all. Chickpeas had very, very low levels, but I would say that not enough to actually cause disease. So I think it's just the oospores are surviving a long time and they're not colonizing those crops. We were hoping that maybe some of them, like a faba bean, would maybe act as a trap crop. There's been some indication of that in the literature, but we didn't really see that, although it said there was some yield boosts after faba bean, but I don't know if it's not because it reduced the oospore levels in the soil. I think it's maybe because it left nitrogen behind in the soil that the peas could then take advantage of. That's sort of what the hypothesis we're working on for that one.
Great, thank you.
Just one more follow-up question, if I can, Nelson.
Yeah, sure.
Just with the work that's been going for quite some time, I think you mentioned the earliest work started in 2013. So has there been a collection of isolates that have been built up over time, and how much diversity exists within the pathogen population across Canadian lentil and pea production areas?
Yes, that's a very good question. My colleague Nick Larkin at AFC in Saskatoon is looking at that. Yeah, between myself and then colleague Sabina Benitza at the University of Saskatchewan, we have a fairly good collection. I have about 35 isolates. I don't know how many she has. It's very hard to isolate Aphanomyces into pure culture and keep them alive. But yeah, I think we have a good collection going. Nick is looking into some of that diversity, and it's a thing like they're more diverse than we thought. Even just based on looking at that ITS sequence, there's actually a lot of diversity between the isolates. And something that is a really big interest is whether there's a divergence between the isolates from pea versus the isolates from lentil. So yeah, lots of questions around that.
Looking forward to hear some more from Nick. Maybe we can hit him up for a seminar in the future. But no, thanks for a great talk. Really enjoyed it.
You're welcome.
Thank you. From the chat, somebody is asking about commercial testing for soils for Aphanomyces.
Oh, yes, yeah. So we do in the prairies, there are a number of seed testing labs that do offer a soil test for Aphanomyces. Right now, they are all based on presence or absence. So they can test your soil and say, yes, you have Aphanomyces, but they can't provide guidance on quantification or what that means for disease severity risk. So that's really what and so right now it's like presence, absence. Yes, you have it, just don't plant peas or lentils. But it could be there at really low quantities. So yeah, what the real research pushes is to try to be able to get to sort of a quantifiable test that has meaning in terms of that actual risk of disease severity.
As a follow-up, and there's also another question about qPCR quantification and if the methods have been published.
Yes, they have been published, and there's a few groups that have been working on this. So mine have been published just in the Frontiers in Plant Science that actually came out of the root rot workshop from last year. So they've just been published in 2023. And then there was a couple groups from France as well that have published methods, and all the methods are very similar based on that ITS gene copy number. So yeah, okay. I can pass on information to the person that's interested if they want it, if they get in touch with me. Yeah.
Okay. Finally, a last question from the chat. Any tests regarding vertical transmission? And there's seeds in brackets.
Yes, yeah. We haven't seen any evidence of that. I get that question a lot from producers, particularly if we have like seed growers and then they want to sell their seed and they had Aphanomyces. So we have tested for presence of Aphanomyces on the seeds and have never found it. So I think it does stay below that soil line. So there's no concern about vertical transmission. What can happen though is if you have lodging, and when you have root rot, plants tend to lodge, then you have to pick up some soil with the plants as you're harvesting them. And so you can get soil tag on the seed, which can have some of the Aphanomyces spores, but there's no actual like transmission from roots up to the foliage. Fusarium is a different story. That's something that we're actually working on right now too, is looking at that vertical transmission of Fusarium avinaceum, because that can happen, but it doesn't happen with Aphanomyces.
Okay, more or less the chat, all the questions were done. I have a question for you also. So what could be the role of phenomics in these applications? You started saying that grading the root disease could be difficult, could be a source of noise in your analysis, and also you showed these nice pictures of fields where in some area there is or there is no the presence of the disease that could be reasonably graded with some, typically drawn data, something like that. Do you have a comment on that?
Yeah, no, that's a really good point, and that is really our biggest challenge, is rating all those diseases, the severity and the subjectiveness of the rating scale. So I think phenomics can have a really important role in this research, particularly when we're starting to screen for and look for resistance and be able to actually quantify what the disease severity levels are. So we have, just as part of the pulse cluster that I just put up the slide at the very end, we are collaborating with a phenomics researcher who is going to be looking at using hyperspectral imaging to see if that's if we can use that to detect what's happening in the roots before we see disease, and then also to screen for use drone imaging.
You mean like images like from Sentinel or from something like that? From
satellite?
Okay.
Yes, yeah. He knows more about all the type of imaging.
Yeah, sure.
But
okay.
Yeah, so we are trying to look at whether we can use sort of this high-tech, high-throughput imaging to correlate with what we're seeing on the roots without having to do the actual root rot, digging up the roots and doing the root rot rating. Yeah.
Which seems very time-consuming. Okay, so this was it. I guess that if there's no more question, it's time for us to thank you again, Syama. This was is part of the ELS webinar initiative, and has been possible also because of CREAMA Institute landed the infrastructure. So thanks to everybody involved, and I wait you all next month with Lars, which was was talking before and asked his question before, again about Ascocita, blight, and lentil. Okay, thank you all, and bye.
Thank you, everyone.